All right, we're going to get started. It's my pleasure to introduce Ron Anafi. He's an assistant professor of medicine from the Division of Sleep Medicine from the University of Pennsylvania. And he's here for a meeting. I'm really glad that you can stay with us in Seattle and give us a talk. He's the last major outside speaker for the Grand Round series of this academic year, so we're really pleased to have him. He did his MD PhD at University of Washington, and he received his PhD in mathematical modeling. He did his internal medicine residency at the University of Vermont, and he started his fellowship in the sleep division at University of Pennsylvania in 2008. Following a year of clinical training, he joined the lab of John Hodgnesh. I think I mispronounced it.
He's not listening, so it doesn't matter.
Working to develop and apply informatic techniques for genome-scale data in sleep and circadian biology. He worked with him to use machine learning to identify new components of the circadian clock. In addition to his work using computational and systems biology techniques to examine the molecular biology of circadian rhythms, Dr. Anafi also worked with Dr. Alan Pack to explore the influence of sleep on gene transcriptions in peripheral tissues. They have demonstrated that large fractions of the heart and lung transcriptomes are influenced by the sleep-wake cycle that are independent of circadian time. Transcripts are induced by sleep in the heart, lungs, liver, and brain, and this is strongly enriched with a metabolic focus. His recent research is focused on development of animal models to understand persistent sleep and circadian disruption following shift work. He's working on developing a clinical test using transcriptional signatures to quickly assess human circadian phase. Working with Dr. Hodgnesh, he's also now began using machine learning to identify candidate molecules involved in modulating sleep homeostasis. And today he's going to talk to us about detecting and understanding the circadian rhythms of human transcription. So thanks for coming.
Thanks, Leo. Thank you guys very much for having me. I'm going to make one small correction, which is I didn't go to the University of Washington, but that's not because I didn't try. This is where I really wanted to go for my MD PhD, but you guys said no. So hopefully today I'll convince you that was kind of a mistake. But maybe not. Maybe you'll think that you made the right decision 20 years ago. We'll see. So that's a long title. Normally I'm so happy to be done with these talks that I forget to acknowledge the people who do most of the work for me, so I'm going to start out with that. So John Hodgnesh mentored me during my postdoc period, and I still collaborate very heavily with him, and in particular now I co-mentored a graduate student named Ray Zhang, who's back in it. He's an MD PhD, and he's back in his medical field, and you're going to see some of the work I did with him. Halil is an associate professor in the University of Koç in Turkey, and somebody else you'll see, I'm a mathy guy, and so I need good biologists to work with me, and he's one of the good biologists. And finally, Junyoung Kim, who's one of the smartest folks I know at Penn, and generally what you'll find is all the good ideas are theirs, and all the things that are messed up are mine. And these are the folks who give us the money, and in particular, I'm primarily funded from DARPA, a large group of us who work on understanding time across biology and across species. So, let's see if this works. I think I might be stuck. Not responding. Restart?
Help is on the way.
Help is on the way. We'll restart. So I'm going to keep talking. So what I'm going to talk to you about, since we don't have too much time, I guess, is circadian rhythms. And I know that's not exactly what you guys do. Oh, one word of sort of in the beginning, say, all questions are welcome anywhere in the middle. So you want to, and that means any question. So Ron, you're an idiot, whatever it is, just say it. I'm willing to deal with it. I've heard it before. So I first wanted to motivate kind of what I'm talking about. So circadian rhythms. So life evolved on this rotating planet. Every day there's going to be light, there's going to be dark. And if you were an organism that could not only respond to that, but anticipate it, you'd be at some sort of selective advantage. You'd know what's coming on in your environment. And if you look at phylogeny, you'll see that rhythms in biology, daily rhythms, are incredibly common. There are a couple of things that probably don't have it. There's some cave fish that have been around for centuries and not exposed to light. Some people say reindeer are far enough north that they've lost it. But by and large, pretty much everything's got circadian rhythms. And it's not just, and it's still not working. Please don't run that fast. So So by and large, everything's got these rhythms in biology, and it's not just disease biology that's a problem. It's not just normal biology, it's diseases. So if you think about it, there's all sorts of diseases that have daily variations in symptom severity, and in fact, these days we appreciate that when you mess up those rhythms, you're probably causing trouble for yourselves. So at this point, shift work is actually classified as a probable carcinogen. So primarily for its increased risk of breast cancer, but all sorts of internal cancers. And there are mutants in circadian timing that, again, have all sorts of problems. So a quick summary, where am I going to go with this? So for those of you not too familiar, I'm going to give you a little review of circadian biology. In other words, the why should I care part of this talk. And then I'm going to tell you two different stories. The first one is most of my postdoctoral work, or a big chunk of it, which is trying to identify the things that drive the molecular rhythms that are going on in biology. And then next I'm going to talk to you about the more research work that I've been doing since I sort of started my own lab, which is really just me, which is trying to tie that, trying to get some actionable knowledge that we can use to influence people or drive drug trials or things like that. So trying to do something useful. Okay, sorry about that. No, it's all cool. It's all cool. So I'll give a little review about circadian biology now that you're really interested. Our story starts with this guy, Androthanes of Thasos. So he is a naval general going around with Alexander the Great. He goes down the Euphrates, and he documents the fact that the tamarind tree, he's sort of documenting everything he sees in this world they're conquering. And he sees this tamarind tree, and he happens to notice that the leaves are closing up and opening with the day and night. But he just thinks, it's just responding to the light. And circadian biology sort of gets a little more of a kick in 1729 when de Marin, who's a French biologist, starts studying the mimosa plant. And some of the mimosas have yellow flowers, which is where you get your drink name from. But if you touch it, you'll see that the leaves close up. But not only do they do that at nighttime and at day if you don't touch them. And at the daytime, they open back up. And mimosa just took them out of the light. And so that they kept on doing that whether or not there was light or not. So they're not just responding to the environment. They're anticipating it. They know 24-hour rhythm, something's going to happen. Now, we're not plants. We're also not these guys. Anybody know what these that little circular dot is there? Little thing red? Neurologist.
Solar panel.
Good answer. So I should know this. I was with my kids at a sort of environmental thing, and they asked us this, and I was going to raise my hand, and my daughter quickly told me not to answer. So, but that is a hole in its head. So that is right above its pineal, a very thin spot where light can enter and directly access the pineal gland, and so they're light responsive. So despite all claims to the contrary from my wife and family, I do not have a hole in my head. Instead, I, like most mammals, work in a slightly different way, and that is the retinohypothalamic tract. So there is a sensor. So light comes into our eye, hits light-responsive melanopsin-containing ganglion cells. They're not involved in visual photoreception, conscious photoreception, but they do detect light. And then there's a retinohypothalamic tract that brings that to the suprachiasmatic nucleus of the thalamus. These small paired nuclei, about 10,000 neurons. And in some very elegant work by Mike Menaker, he showed that that small neural structure really controls these behavioral rhythms. But the modern biological era became with this guy named Seymour Benzer and his grad student Ron Konopka. And they were working in the late 60s, early 70s, where everybody thought, you know what, it's all environment. That's what's controlling things. We're kind of hippies, looks pretty good. And they weren't really interested in sleep and circadian rhythms. They just wanted something that was a very identifiable output of behavior that they could study. And that seemed to be rhythms in animals. And they looked at the Drosophila eclosion rhythms when they leave the pupa. And they identified in the mutational screen 3 mutants, or I think 4, that all sort of had alterations in this. And when they went back and mapped it, they all found out that they were alterations in the same genomic region. And years later, when they cloned that, that was what we call the period locus. And in mammals, the stuff really began there was the genetic period in mammals really begins with Joe Takahashi's work, which is ENU mutation screen where they took mice, and just so you see what's going on here, so they're on a running wheel, and this is the black is when they're running, and the light is when they're not running. And here there's light dark. The black barks are when it's dark, mice are nocturnal, white are when the lights are turned on. And you can see that the mites start running when it's dark, and they stop running when it's light. And they keep on doing that, and then you turn off the lights. And this is DD. And you can see that they keep on having a pattern. Each day they shift back a little bit, so it's not 24 hours. It's 23.9 or something like that. But they're keeping a very good pattern. But then they found this mutant. And they noticed when they turned the lights off here, and this was a heterozygote, it gets later and later each day, and quite a bit. And then if you have a homozygous mutant, you turn off the lights, and you can see that the circadian timing is effectively smashed. It's not working well at all. There's no discernible rhythm and activity. And so this idea that a single gene, when you mutate it, can disrupt this very clear behavioral pattern was amazing. But one of the things that's really allowed circadian biology to move forward is that it's not just in your brain. If it was, it would be hard to do this molecular biology. And each time you make a perturbation, you have to wait for the animal to grow up, study its behavior. Turns out that the clock mechanism is conserved in almost all of your cells. This is some work from, this is a more recent paper from Andrew Liu and David Welsh. And you can see that if you have a reporter that's showing the activity, these are skin cells of fibroblasts. And they're maintaining the same rhythm independent of a neural architecture, not even neurons. And so because it's so ubiquitous in things that we can play with, we were able to get a much quicker version of idea of what's going on. So here's the core clock machinery. A simplified version. There are these two genes, clock and BMAL. And they come together and they induce the transcript, they bind to an E-box element on the promoter of lots of genes, and they induce the transcription of those genes. And very prominent among the genes that they transcribe are cry and period. And those genes get transcribed, they leave the nucleus, they come back together, and then there's a delay, and they have to be imported back to nucleus, and they turn themselves off. So imagine in this room, there's a thermostat that's not working very well, and it always tells you the temperature 3 hours ago. So you set the temperature at 70, and it says, oh, it's not 70 yet, I better turn on the heat. And it gets hotter and hotter, and it hits 70, but the thermostat doesn't see that. It's still seeing the temperature 3 hours ago, and it gets hotter and hotter and overshoots. Finally, 3 hours later, it says, wow, I way overdid it. Better turn on the coolant. Goes the other direction, and it overshoots. So this mechanism of a feedback loop with a delay induces oscillations, and that's what's going on in biology. But as we've sort of progressed, we've seen that there's a lot more than that simple loop. This is a little bit more of a contemporary slide that's showing there's this core loop that I just described. Clock and BMAL also influence a second family of group genes called the revs and the roars, and they sort of perhaps stabilize that loop. And then there are things that are adjusting the degradation, so all sorts of things that are setting timing. And no less an authority on health than last week's Wall Street Journal. So if you look, this is just what they actually, the graphic that they had last week. Because this clock is not just in your brain, but in all sorts of your body, you can expect there's lots of biological consequences to time. So blood pressure sort of peaks. Blood pressure, core body temperature, and heart rate are at their lowest in the overnight. In the early morning, cortisol and stress hormones keep going up. Heart attacks and strokes are peaking somewhere between 6 and 12, 6 in the morning and 12 in the afternoon, and it's a pretty strong predominance. They start having other things. Epilepsy syndromes, in particular temporal lobe epilepsy, starts to happen in the midday. Of course, nocturnal frontal lobe epilepsy, probably more to do with sleep patterns and circadian, happens at night. But lots of diseases are having variability as a result of these patterns. And probably in medicine, the way that this has been most studied is in cancer chemotherapy, where some of the chemotherapeutics, particularly the older ones that were studied, had as much as a twofold difference in toxicity responses, depending on when you gave the drug. So I sort of talked about a little of this. I'm going to sort of skip it since we've wasted some time. So now I'm going to tell you a little bit about some of my older work, which is I've tried to convince you those rhythms are important. How do we figure out what's driving the rhythms? What are the core nodes of the pathway? So I already told you that there's this core clock network that we already know about. And if you look it up in a book, you can look up core clock network. It'll tell you 15 or 20 genes, depending on the book you look at. And they've been the focus of a lot of biomedical research. They've been independent of timing, implicated in obesity, metabolic derangements, all sorts of things in cancer. But they've resulted from lots of years of very careful work. Typically, these mutagenesis screens mutate a lot of animals, figure out, find one that's got an aberration, go back, do sort of breeding, clone it. But we also know that picture is incomplete. if you look at assessments of how much of the variance in circadian rhythmicity in mice or in humans is explained by the known clock genes, a small fraction. If we look at the mathematical modeling of how this works, we know there are loops that aren't well explained, that they have to put in lots of fudge factors. So the question is, can we try and fill up this picture, and can we use the wealth of information that's already available to do that? So if I'm going to tell you I want to find core clock genes, I better tell you what I mean. And that's not so easy to do. So what's a core part of that watch? It's not going to work without, if you don't have the band, it's going to fall off of you, and that's not so good. But you might not say that's a core part of the machinery. A battery of your watch, everything dies without the battery, but you don't think about it as a core part. So we went to circadian biologists and we said, what do you think makes something a core component of the clock? And they gave us these criteria. One, core clock genes cycle. That is, their expression over the course of the day is changing in a 24-hour period. They impact circadian phenotype. You knock them out, you mutate them, you should see a change in behavior. They interact with each other. They're part of a functional mechanism. They're ubiquitously expressed. So in all parts of the body, we know that this clock is running. So if it's a really core part of a loop, I should find it in a lot of places when I look. And they're conserved through a phylogeny. So remember, Drosophila had a pretty similar clock. Lots of animals do, so we should be able to look. Problem is, none of those rules are absolute. I can find a gene that violates each one of those rules, and in fact, if I look at each gene, it violates one of those rules. But each one of those rules, each one criteria gives us some evidence. And our goal is, can we use all the information we have, combine the evidence, and find high probability candidates? So this is a lot like what we were all trained to do in evidence-based medicine. So you see a patient, you want to know if they have pneumonia. You might say, are they coughing? You might say, okay, they are, they're not, I'm going to sort of pursue this. I might get a white blood cell count, and I might know what the distribution of normal white blood cell count is in the population, and I sort of know what it is with people with pneumonia, and I can say, how much does that shift my evidence? And then I might get another test, an X-ray, and again, it shifts my evidence each bit. And as I add bits of evidence, one way or the other, I can make a decision. You also know this in terms of Google filtering your spam. So you might actually have a rich uncle who's visiting Nigeria, and you're very close with him. So he told you that something that he forgot his Viagra prescription, and he wanted to know your bank account number because he wants some funds. So that's all possible. But each one of those words is increasingly unlikely in normal mail. And so Google keeps track of how frequently are those words in normal mail, and they add up the probabilities, and when you hit a certain threshold, it puts it in your spam box. So we want to do the same thing. We want to evaluate the post-test probability, knowing the pre-test probability. In other words, we know what the population is of white blood cell counts in the population. We know what it is in people with pneumonia. Can we start figuring things out? So here's our recipe. And identify some metrics, some number I can quantify about each of those features that I told you about, and then figure out how it's distributed among all the genes in the genome, and look at our examples, the things that we already know are implicated in the clock, the people who have pneumonia, and say, how's it distributed there? Then we're going to compare those two and compute some sort of evidence factor. How much should it shift my decision? And then we're going to add up all those bits of information. So first bit of information I said is it needs to cycle. So there have been a lot of experiments now that have looked at what's cycling in different tissues. And so we very simply took a bunch of tissues. Actually, this is an earlier version, so it was only 4 tissues at the time. And for each gene in a given tissue, they assign a p-value. P-value 0.01, it's cycling, 0.02. P-value close to 1 means it's probably not cycling there. And we just very simply multiplied them up from several tissues. So a very, very small number means it's cycling robustly in many tissues. A number close to 1, or a log number close to 0 means it's not cycling at all. And if you look at the entire population of genes, you can see most of them have, if you take the log of that number at 0, in other words, the number itself is 1. But if you look at the, let's say, 20 genes that I used as examples of the clock components, it's very different. And so then you can ask the question, if I meet a gene named Bob on the street, and its number was right here, how much should it shift my evidence? And you can compute in a sort of Bayesian way that number by comparing those two distributions, and that's what that is. I can't knock out every genome in the genome, but pharma can do it with siRNA. They can knock down cells in RNA molecules of cells in a dish, and that's exactly what they did. And they actually found hundreds, if not thousands, of molecules that modulate rhythms, which is why it's a little hard to use that data in isolation. But if you look at all genes in the genome and you give a measure of how much it messed up their rhythms, you see most of them had a very modest effect, but there's a long tail. And if you look here, you can see a very different distribution for the genes that we know about that are involved in the clock. And you can compare those two and you come up with some evidence factor. You can do the same thing in looking at genetic interactions, compiling a lot of information. Do these proteins interact in known databases? And you can put it all up, and I can rate the genes in the genome based on their clockitude, how much it seems like they contribute. So we put in some example genes, clock, MPAS2, BMAL. This is the official name for BMAL, the period genes, the cry genes. And then we rank the genome. And good sanity check, you get back a lot of the genes you put in. But you also get some new candidates. Some of them are involved in the cell cycle, PSEN2, so implicated strongly in Alzheimer's disease. Some genes that are known to be involved in the clock and I didn't tell it about. And then this gene called GM129 that I'm going to tell you about. But you might just say, well, you know what? You put in the information about this. Am I getting any new knowledge from this? Does this mean anything? So we can play this little parlor game. We can pretend I never learned about clock, leave it out of the equations, note, and then say, wow, if I use this approach, would I have found it? And then I can do the same with another gene and leave it out. And we took out leaving 2 of them, in other words, 10% of it, at a time, and tried to leave out every pair and say, how likely would you have been to find this gene using this method? And that's what this cross-validation is called. And you can see this is how far down the list you go and your chance that you'd find the gene. And it boils down to is if you look at the top 50 candidates, there's a 50% chance you would have found one of these clock genes. Now, it's not perfect, but that's a big advantage from having to screen the entire genome. So GM129 was one of those candidates. And when we looked, we found it not only cycled in the 3 tissues we looked about, it cycled everywhere. In fact, at this point, it cycles in 14 tissues that we've looked. And it physically interacts with the known components of the clock, or at least 2 of them. So this is a mammalian 2 hybrid scheme with the 2 bits of a protein. There's an activating sequence and a binding domain, and those are both fused to different proteins. And only when they come together do they induce the transcription of a reporter, in this case, luciferase. So if 2 proteins bind, the lights come on. And here you can see that we compared lots of different clock genes against chrono, which is what I call GM129. And you can see it bound per2 in BMAL. And we confirm that with normal pull downs. But not only that, did it bind it, it functionally affected the clock. So I told you that clock and BMAL induced the transcription of a gene called PER. So here we have a PER luciferase reporter. So it's got some basal expression. If you overexpress clock and BMAL, it goes way up. If you overexpress chrono on top of that, that represses it. And the canonical repressor is cry. That's here, so you can see it repressing. Here's chrono repressing. So it's about as effective in terms of the overexpression in stopping clock and BMAL's activity as this canonical repressor. And in fact, if there's a point, we know about certain point mutants of BMAL and clock that are relatively resistant to cry. They don't stop chrono's act. So it seems to be working in a different way. And this is the siRNA experiment, the reduction experiment, it's the opposite. And more importantly, if you knock it out in an animal, you see that you also change its rhythm. So this is the wild type animal, which has a 23.2 hour, and this is 23.7. In general, it's about a 30-minute difference between the two. That's the same as you'd see with a period knockout or a cry knockout, several of the other known clock genes. But it doesn't affect the light response. So we called it a computationally highlighted repressor of the network oscillator, which goes for chrono, in large part just for propaganda purposes. And this is Chronos, the sort of personification of time in Socratic philosophy. But the question is, how's it doing what it's doing? So we needed some hints. We needed some good biology there. And the way that we started looking at that was that chrono is BMAL specific, BMAL1 specific. So there's BMAL1 and BMAL2. They're almost identical genes. But chrono is binding BMAL1, but not BMAL2. And its functional repression was there for BMAL1 and not BMAL2. And that's what I'm showing down there. So we went ahead and tried to identify the important real estate. What's the key differences between BMAL1 and BMAL2? And if I can say it's working in one and not the other, I can start making these chimeric proteins and say, where's the area of the gene that's important? And I'm trying to rush through some of this. But it turned out that we identified a region at the C terminus, at the very end of BMAL1. And that domain already had a function. It found something called CBP, something that activates BMAL. So we had this hypothesis. Oh, now it's my problem. We had this hypothesis that that chrono was abrogating the interaction between BMAL and cry. And we went ahead and showed that with sort of molecular fluorescence, and that in the presence of chrono, the interaction between the normal interaction between CBP and BM129 and between CBP and BMAL went away. So a sort of mechanism for how this is working. And I also wanted to tell you, and so we described that. At the same time, a Japanese group found it a different way, published it together. And the approach didn't just yield GM129. And one of the things that I'm presently doing is sort of tracking down some of the other things. So an interesting finding that I'm just going to mention briefly is this gene called CBS, which is cystathionine beta synthase, which is an enzyme which you'd never think would have much to do with the clock. And it catalyzes this important part of transsulfuration from methionine to homocysteine. And so it regulates glutathione and cis-aldomet, things that are methylating proteins or DNA. And it's one of the two enzymes that makes hydrogen sulfide. So it's got a lot of interesting properties. I don't know how it's working. And it's implicated in a lot of disease. But the main point is that CBS binds cry, CBS potentiates cry, and cry in turn potentiates its enzymatic activity. So they come together as an enzyme, it works better, and cry is working better. We're trying to figure that out now, but I will tell you that animals who are knocked out also have messed up rhythms. So just a little, not going to tell you the story there because we're trying to work out the biology, but this idea of integrating data to accelerate biology and learn something can be really useful. So interim conclusions, this is sort of book smart machine learning. You take what doctors already knew was important, feed it into a computer, help it guide our experiments, can accelerate our progress in biology and in circadian science. But I want to move towards something more medically relevant and towards something that we can think of as actionable. And that's what I've been working on later. So I will tell you that I think we've been looking at rhythms in all the wrong places. So I've told you that circadian disruption is a risk factor for several diseases. Clock genes are disrupted in lots of cancer lines. Is it disrupted in real cancer? I have no idea. I told you that it's probably involved in lots of drug metabolism and drug targets are cycling. Which drugs? Which time? I don't know. Which diseases change which rhythms? Because that's supposed to be true too if you look at the circadian literature. Which ones? I don't know. So why don't we know? What are the obstacles? Oh man. So the obstacles are this. The typical the obstacles are it is hard to get data from people. I normally get a circadian a sample once an hour from a mouse. I can't get a person to volunteer to give me a slice of their liver every hour. And even if I did, the IRB wouldn't let me do it. So even harder to get cancer patients to agree to let them have a slice of their tumor once an hour. More to the point, mice, very clean, very little difference between them, inbred strains. People, lots of variation. So I need lots of data, lots more data to determine rhythms in humans than I do with people. So we need to figure out some way around this. And the question that I had is, can we identify the output rhythms in unordered data? So if I don't synchronize people and do all that stuff, if I just have a whole bunch of data from all sorts of different times and I don't know what the time is, can I figure out what's cycling? And can I start making some clinical conclusions? And if we can, there's lots of possibilities, because while it might be hard to collect samples in that sort of regimented way, there are thousands of patient biopsies from normal tissue and from cancer and all sorts of things. So if we can do that, and it seems like a hard question, then maybe we can make some progress. So here's the underlying idea. Rhythms leave a trace. So imagine I had two genes that are oscillating. Let's say clock and period, that are out of phase with each other. When clock goes up, period is maybe midway through. As it continues to go up, the other one starts going down, and clock goes back down, and the other one goes up. And if you would plot one versus the other for time, they'd make that little spiral. Now, if you didn't know the temporal order, and you just shuffled this, it would look like this. It wouldn't look like a spiral anymore, but it'd lie on this cylinder. You'd see in there, and you could look at it and say, wow, I still see some structure to that. And in fact, if you just didn't even plot time at all, or this random number, which is there at the bottom, they would make a circle. And even if I said, okay, I have a point that comes from this angle of the circle, I would say, wow, it comes from here or here or here, this time of day. I might not know what day it comes from, but I can at least figure out what time of the day, what part of the cycle it comes from. So biological rhythms leave a big trace. If I look at any tissue, there are not 2 genes that are cycling, but the estimates are around 10% of the genes in any tissue are cycling, so thousands of genes. So if I did that same experiment with 1,000 genes that are cycling with the same period, they trace out this circle in a 1,000-dimensional space. Kind of hard to find and picture, but something that we can look for. So I'm going to try and find the circle in this high-dimensional space that best matches the data, and then assign each biopsy sample, each piece, some phase, and then go back and figure out what's cycling in human beings in different pieces of tissue. So here's some first playing with numbers before we go to real data. So I made up some data, had oscillatory patterns and exponential patterns, a whole mix of things. And then I plotted the eigengenes, that's the principal components. And you can see there's one that's just going up, one that's an oscillation, things like that. But if you project it in the right way, you would end up finding a circle. If you shuffled it, that doesn't change anything. You still find the same circle. But if you had some sort of data that didn't have any oscillations, I can't, no matter how I try, make a circle. And that's the point. So we can take some real data. So this is the same idea. These are the top 2 eigengenes, the principal components. So your question, we'll get to that in a little bit. So your question is, what if I don't have surgical biopsies are collected during the day, not the night? I'm not going to have an even distribution of points around the circle. Absolutely true, and that's going to be one of the things that I'm going to have to deal with. So this doesn't have that problem. So this is a mouse experiment where they took liver every hour, and they did it for 2 days. And then I went back and tried to find the best circle I can. And then for each point, I said, what's its angle on the circle? How does that compare to the true time at which it was drawn? And that works quite well. So the question you might ask, oh, this is not my day, is how do we do this? And at this point, you cannot believe anything I say that has to do with computers because mine keeps crashing. So I'm going to keep talking. So I use the same trick that Google does when you search for cat picture. So that works surprisingly well. But there is not somebody whose job it is to say cat picture, not cat picture. Cat, that would be a good job, probably better than mine. But they don't have anybody that they employ to do that. Instead, they have something called an autoencoder. And it's a neural network. And a neural network is a very... I turn off your program because it doesn't look fine. So I might skip it. So the way it works, never had this much problem. I'll be right with you guys. Is a very simple thing. You have some inputs to the network, and it connects to another neural node. And this node has all these inputs, and it adds them all up. It says, if this one plus this one plus this one is over some threshold, it fires. And you add them all up, and you can have all sorts of architectures, and then you have some outside. And you ask this to do what seems like a really stupid thing. You say, I just want you to reproduce on the outside what I put in. And you go, well, I could have done that without a neural network. I just draw the same thing. But you make it hard for the computer, because in the middle, you constrain the number of bits of information that it can keep track of, so that it has to somehow compress this data. It has to keep track of it in a way that is meaningful but keeps less information. And in the middle, I make it compress onto a circle. So I say you have to compress all this genomic data but you can only keep track of one phase angle on a circle. Find the best way to do that. And then we can ask the question, does this genetic data that we see, is it better fit by a circle than some random data that I created with the same parameters? And we can assess the significance of that. Say, does this really look like it's been generated by something that's circular? And if it does, then we can assign every bit of data a phase and see what's cycling. So there are going to be some problems with human data. One of them is it's not going to be uniformly collected, which we'll see some of the bits of that. One of them is there's a lot more noise than there is for mouse data, and I'm going to do my best to minimize that. So I already know in the mouse 1,000 genes, let's say, in the lung that I think are cycling. Same exact 1,000 aren't probably going to cycle, but those are probably very enriched for what's cycling in the human. So I'm going to keep track of it in the beginning. There's those 1,000 genes. And this time, my estimate of time based on that. So here is some data that I took to start. This was about 1,500 biopsies of human lung. 500 obtained in one site in Canada, 500 in another site in Canada, and 500 in the Netherlands. These are honestly people who are sick. Most of them had cancer, but this is the normal lung adjacent to it. And we did this same profile, tried to assign each biopsy a phase based on its role in the circle, and then plot out what gene expression is doing based on those phases. And so this is some of the core molecular oscillating, clock, cry, reverb, beta. These have the same phase relationship you'd expect from the mouse. And these are output genes that canonically are all expressed at the same time, and you can see that in the person too. Now, you might have some questions, like one of them is there's not even, so you can see a whole lot of points here, much fewer points there. And in fact, well, let me get to that. One problem that I had is I plotted out many of them. There were 2 probes for clock in the exact same opposite pattern, and you can't believe that. And it's because one of the probes was close to dead and has barely any variability. But for the most part, this works quite well. But the question is, what about the distribution of how many samples am I getting at different times of the day? Surgeons are only collecting these biopsies for the most part from 6 in the morning to 6 at night or something like that, being generous. And if you look at the histogram of when we recover, when we say this sample was taken, many more come from one part of the cycle than the other. In fact, about 80% come from here and about 20% here. But that matches very well the data about what fraction of the United States and Canada are shift workers. So there's about 15 to 20% of the population that are shift workers. So if you get a daytime biopsy from their lung, we're getting a glimpse of nighttime biology. And putting it together, we can start to put this picture together. Now you could say, it works, maybe. Can you show me, or is this at all, give me some validation. Of course, if I actually had a gold standard for human tissue, I wouldn't need to do this. So I really don't have that. But what I can at least tell you is that if I'm wrong, I'm consistently wrong. So I separately analyzed the data from Canada and the data from Europe. And we said, if a given gene peaks at a certain phase of the circle in Canada, what phase does it peak in Europe? And you can see this follows very well the line of identity. There's clearly some noise. But we're getting a very consistent answer about what genes are cycling and when in the day they're peaking. So how do you use that? Well, one of the ways is for drug timing. So I told you there are lots of drugs that maybe have circadian modification, but how do you figure them out? Can you test every drug? Well, if you take simvastatin, a cholesterol medication, your bottle says to take it at night. And the reason it does is when Brown and Goldstein were studying cholesterol pathways, they actually saw that human beings are synthesizing the cholesterol at night. They had indwelling catheters and things like that, and they were aware of that data. So if you take a long-acting medicine like atorvastatin, it doesn't matter so much. But simvastatin has a short half-life. And they said, when you test this medicine, you better test it at night. So now we can sort of do that same sort of thing. We can have a hypothesis-driven search for what medicines, in the lung, for example, are going to have day and night regulation. In fact, if you look in the mouse, a lot of medicines that we take sort of cycle somewhere in the mouse, but we don't know if they cycle in the human or in the right tissue. But in the human lung, ACE cycles. Now we think about ARBs, and ACE inhibitors for blood pressure, but ACE primarily acts in the lung, and that's where that conversion from renin to angiotensin 1 to angiotensin 2 takes place. Beta adrenergic receptors cycle there, and various immune and growth factors that are being targeted for IPF. So this might tell us, wow, you should time that trial. Can't do that for every trial, but you can, for some, if you have a good reason, you can time it. So another thing we want to do is say, wow, can we learn some biology from this? So that's actually kind of hard when you have thousands of genes changing to learn something, so get some cohesive picture. So working with a grad student named Ray Zhang, we came up with a way of doing this. So this is very similar to something called gene set enrichment that I'm sure many people have seen, which is if you have some pathway that's composed of many, many genes, and you do some perturbation, let's say compare smoking to non, you can did those go up and compare it to the population and compare this one set of genes to everything else, where you already know how that set of genes is related. Problem is it doesn't work for periodic things because 0 is very close to 24. When you get very high, it comes back to being very low again. And so we had to just, not a giant change, but rewrite the program so that it works for periodic things, for cell cycles and for circadian rhythms. And the way it works is you end up totaling, let's say you have 0 to 24. Every time I see a gene that peaks at that time, I might raise it up, and this is a histogram of probabilities. So starting out at time 0, no genes have started peaking yet, and by the time you hit 24, they've all peaked. And you compare this cumulative probability distribution to just a uniform distribution. And you add up this big positive discrepancy and this negative discrepancy, you add them up and you get some statistic. And if you can do that, you can start seeing what pathways are overrepresented. First, we did this with lots of mouse tissue and have published an atlas about that. But we can start looking at the human lung and saying what physiology in the human lung is happening at different times of day. So this is a very busy slide, but showing you, for example, latent infection to Mycobacterium tuberculosis, peaking right about there. And you can look at some of these things. So here is the tuberculosis pathway. So these are the genes that are peaking. You can see right a bunch of daybreak, midnight, a very strong clustering. So if you wanted to treat somebody with TB, you now have some evidence to say maybe I should target my therapies for certain times of day. There's also the mycobacterial rhythms that we're not talking about, but in terms of the host response, it's going to be strongest at certain times. The immune system, cell cycle are also showing variabilities. And we can start making some educated guesses about what therapies to target, what therapies to look for time signatures, and sort of move this field a little bit forward. So there are some clear limitations to this. So in all of this analysis, when I make these circles, I actually don't know that it's cycling with time anymore. If I get out these circadian genes that we know cycle with time, that's a good indication, but it could be that they're cycling with latitude or some weird thing that I know nothing about. But to the extent that you recover circadian genes when you do this process, it's at least reassuring. We also know that I'm working with transcriptomic data right now. That ain't protein. And drugs target proteins. So there's always a little gap there. Love to use this on proteomic data, but it's just not that available yet. And there's a danger of circular logic. In other words, I tried to find some ordering that made things the most circular, and then I found it. So you can always question, did I just put that in? And there's some elegant ways of dealing with the statistics to try and overcome that. But no matter what math I show you or even show myself, it's always going to be a little bit of a worry. And then there's a question of statistical versus biomedical significance. So when they've done these circadian rhythm experiments before, they have 50 samples. And in order to detect a difference, these are generally things that have high variability in amplitude. I have 1,000 samples, 2,000 samples. And we can start detecting very small oscillations. The questions are those at all physiologically meaningful. So right now we're limiting things to things that at least have a twofold amplitude. But the question is, got to start looking about what's physiologic and what's just stuff that we can get out of the math. So the next steps in this, we are creating a database of human circadian rhythms based on tissue biopsies, starting with liver and lung and fat, things that there's enough of them that we can validate, show that we got the same answers in different when the data is obtained from different places. We're trying to identify rhythms in disease samples, so in particular starting with lung adenocarcinoma and saying how do the rhythms change? Can you target therapies in that way? And finally, looking at allelic variants that cause expression changes in time. So not just, as it's been done, saying, is there some allelic variant where HMG-CoA reductase is expressed more in some people than other, the cholesterol, but we know that gene has a 5-fold oscillation over the course of the day. So just finding that is going to be hard, but if you can adjust those things for time, then you can start going forward. So hopefully I've shown you that this is a powerful tool, or at least a coming tool, to identify the rhythms in human transcription and human tissue, and that when we couple that with phase enrichment, looking at pathway enrichment, and looking at drug targets, we can start making some hypothesis-driven leaps forward in chronomedicine, which at this point has really just been some people happen to notice that time made a difference here. And until you move beyond that, it's hard to make any sort of significant progress in the field because we can't test every drug, but we have to have some ideas of which ones are worth testing. And that's what we're trying to do here. It's also worth pointing out that these same kind of methods can help you figure out where your patient is at any given time of day. So if we take a blood draw from a person and we then know what time that was drawn, we can do the same sort of thing. So here, these are blood draws, red, yellow, green, blue, purple follows the color wheel. That each of those is a 6-hour window, 4-hour window. And you can see that we can start tracing out from a single blood draw what time of day this was drawn from the person's circadian biology. Right now, trying to figure out what circadian phase a shift worker is in, or people in general who have sleep disorders, is actually quite hard. We have to bring them into a lab, get rid of light, measure melatonin in a very regimented way, and we never do it. And so to do studies about how phase and circadian rhythms are affecting some outcome are practically impossible. But if you can start having blood tests where you can measure those things with a couple of hours of resolution, then again, you can start moving the field forward a little bit. So I thank you for your attention and for putting up with my various technical difficulties. And any questions are welcome.